Hey, Craig.
Hi, Mike.
Just about to get started here, Craig. Just waiting. It's just gone 12:02, so maybe we'll get started. I'm going to start off by introducing our two speakers today, Dr. John Tyson and Dr. James Zlosnik. Is that the right
Mhm.
pronunciation?
That's
Beautiful.
good.
Okay. So Dr. Tyson has a background in molecular cell biology and genomics. Particular focus is on next generation sequencing technologies and molecular diagnostic. He joined the BCCDC as a senior scientist focusing on microbial genomics in 2021 and the Department of Path and Lab Medicine at UBC as a clinical assistant professor in 2022. In his current role, he helps oversee the developments of the microbial genomics portfolio at the BCCDC Public Health Lab. Ongoing efforts include standing up diagnostic assays and next generation sequencing across a wide portfolio of public health related pathogens. And Dr. James Zlosnik leads bioinformatics for pathogen genomics at the BCCDC Public Health Lab. He has more than 20 years of research experience and over a decade applying microbial genomics to public health. At BCCDC, James oversees the development, validation, and operational maintenance of bioinformatic workflows for diverse pathogens and has helped expand the test menu to include Mpox, influenza, healthcare acquired infections, as well as TB drug resistance prediction and HCV genotyping, two of the first accredited clinical genomic tools in Canada. He is also a clinical assistant professor in the Department of Pathology and Lab Medicine at UBC. And today they're going to be talking about pathogen genomics in the BCCDC Public Health Laboratory. So welcome both James and John. Really appreciate you coming and presenting to us. Whichever one is going to present first. James?
Yep, that's me. I'll start off. Thank you very much for the introduction. And just to start with a land acknowledgement, just to acknowledge that BCCDC Public Health Lab is located on the traditional ancestral and unceded territories of the Squamish, Tsleil-Waututh, and Musqueam nations. And also to acknowledge that I'm presenting this from my home here at Port Moody, which is on the lands of the Coquitlam, Tsleil-Waututh, Musqueam, Squamish, Catsi, Kwantlen, Kaikwat, and Stolo peoples. Thank you for that. And then also, I just want to really start by acknowledging that today we're going to present an overview of the genomics program at BCCDC. Really, I'm just here as a sort of here presenting this on behalf of a very, very large number of people. And I can't really sort of hope to acknowledge everybody in a single slide. And so I'll start in advance by apologizing to anybody who I have missed off here. The range of people required to sort of make genomics happen in a public health context is large. I think the team that we have at BCCDC PHL is just an amazing team of people who all work together in a tight and collaborative manner to deliver the mandate of the Public Health Lab and to deliver genomics over the past few years. I'd just also like to give a particular shout out though to my boss Linda Huang, the medical director of the Public Health Lab, and Natalie Pristajek, who've really been instrumental in setting up genomics in the Public Health Lab. And that is their tireless advocacy and pursuit of genomics has really helped build genomics at BCCDC PHL, as well as all the people who work on genomics on a day-to-day basis and integrate it into their products and teams and other things. So what do we do at the Public Health Lab? I largely work really on the genomics portion, but we can think about the way the Public Health Lab works as it delivers service to perform diagnostic and reference testing. So on an individual patient level, it does that, and it does that for certain key pathogens as a sort of reference center. And it also provides patient epidemiological and outbreak consultation. So for the bigger sort of picture beyond an individual level. And in addition, we innovate through applied research, and that is in part what our UBC affiliations and roles allow us to do. So we have this great center that does this. And I just want to just expand a little bit because it's not always intuitive to people who work in other parts of the healthcare system that we're not only patient focused, although we are patient focused, and individual results are obviously very important to us. But we also have an additional mandate, which is the population. And so we kind of think about this in having two patients. We have the patient, and then we also have a second patient, which is the population. And this is important in pathogen genomics, as everybody will know. Pathogens don't respect borders; they don't respect anything very much, and they spread easily. And we need to be able to keep an eye on those and be able to surveil those and understand what's going on within our own population. So the PHL serves more than just individual patients. And we also have a range of partners as well. So we have local partners, such as internal BCCDC service lines. The Provincial Infection Control Network is another example. Regional health authorities are another key example of our partners and stakeholders. Provincially, we have others, including the Ministry of Health and Ministry of Agriculture, as well as our local universities where we collaborate on research. Nationally, we have partners in the Public Health Agency of Canada, particularly the National Microbiology Lab in Winnipeg, as well as the CFIA. And internationally, of course, we interact with state labs in the US sometimes, the US CDC sometimes, people like the UK HSA and others that are working at the front edge of pathogen diagnostics and surveillance as well. So that's just to acknowledge our partners as well that are key. And the BCCDC PHL, probably this will be familiar to some people, but just to also acknowledge that the Public Health Lab is structured into individual discrete programs that specialize in individual parts of infectious diseases and other things. And they're split out here. And there is a development arm that I'll come to in a minute called molecular microbiology and genomics. And then in addition, we're in the process of hopefully developing a genomics program that will serve all of these programs with genomics products and results. So that's just to give you an idea of the scope of the structure of the PHL. I thought it was worth just taking a quick look back at, or not even look back at, but a quick overview of how pathogen diagnostics are performed, both historically and to this day to some degree. You'll see here that this is from a 2012 review in Nature, and Nature Reviews Genetics, and it gives you an overview as to how bacterial samples in this case are identified and the timelines required for that. And typically a lot of this still stands today, and the timelines in many respects for some of this are acceptable still in terms of at least initial diagnostics. But in addition to initial diagnostic down to what specific species is causing this person's infection, there's also often a need to do further investigation and characterization in the form of typing. And typing goes beyond the strain level identification, but goes to say exactly what is this person infected with and how is it related to other things about them. And historically, that can take some considerable time to do, so it can take 1 to 6 weeks sometimes to do that. But there are some pathogens like mycobacteria too, where their growth rates are very, very slow. And so even an initial diagnosis can take 1 to 3 weeks. And then on top of that, when you want to do antimicrobial susceptibility testing, that can take further time because it requires further culture. And typing can take even more beyond that. And so the timelines for getting an initial diagnosis and perhaps an initial susceptibility pattern for mycobacteria can be in the order of weeks, which delays both diagnosis, but also delays treatment options for physicians too. So while historic approaches are both going to continue and are necessary in many ways, there's also scope for improvement. And this is in the same review, just an overview looking, and this really focuses a little bit more on the typing piece, and also I think the timelines are still a little bit ambitious. 1 to 12 hours is technically feasible for some of these things, but in practical utility, it's larger than that. But it does, you can see that it shrinks down that timeline from months in some cases to weeks, a week at the most, and days more likely. And so this is the advantage that genomics generally offers us in terms of pathogen diagnostics, both speed and also more additionally, it enables a resolution that is just simply isn't possible with a lot of other traditional methods. And if you think back to like the original methods of biochemical testing, give us a very, very limited insight and often not complete insight into even the species. Over time, other methods like typing methods such as pulse field and PCR came along, but they also give a limited insight. Things like PFG don't work at all anyway for viruses. And then there are historic sequencing approaches like MLST, which is a sort of small barcoding approach to looking at only parts of the genome that give you a limited insight. And the advantage that whole genome sequencing, I think, gives us is it gives us the ability to see all of the nucleic acid composition of an organism in a very nearly unambiguous way, and gives us a very high resolution insight into what is making up the pathogen that is of interest. And I think this is clear. I mean, the World Health Organization has published this document highlighting a global genomic surveillance strategy, outlining why this is important and why the benefits of whole genome sequencing for a lot of pathogens. And so it allows things like monitoring of diseases, ultimately control of pathogens through infection prevention and control interventions, and also it's important for developing countermeasures like vaccines. And so this is an important technology that's recognized widely and is being used increasingly widely by a lot of people. So why now genomics in public health? Well, as I mentioned, we get the higher resolution. The cost of sequencing over time has dropped considerably. The additional the bioinformatic tools and other tools that are needed to do this have matured to the point where they are reliable and can be used repeatedly. So those are important too. But I think the other thing just to acknowledge is that SARS-CoV-2 happened, and that has been certainly a catalyst for the adoption of whole genome sequencing in public health labs, and it has facilitated really and enabled this through the acquisition of additional skill sets, which is people, and as well as additional equipment and things like sequencers and things like liquid handlers and things required to do high volume work, as well as computational servers and other things. So the acquisition of these has been critical, and fortunately at BCCDC we have been able to acquire the necessary component pieces to operationalize genomics for a range of pathogens. This is just a quick cartoon just to illustrate when we're talking about pathogen genomics, just to acknowledge that most of the time we're talking about whole genome sequencing. Usually if you can get the whole genome, it's better. It gives you everything and you can filter down to the bits of data that you need. But just sometimes it does also have utility for other things. And one of the local applications we've got for this is HCV genotyping, where we PCR up just a segment of the genome and then we put that through next generation sequencing to get the results out for those. So it usually talks about whole genome sequencing, but it is applicable to amplicons and other things like that. And then the other thing that I wanted to just mention is about pathogen genomics is that it is a discrete discipline. And I think that in the healthcare system, we're often used to hearing about genomics because it is a big deal when it comes to human diagnostics. But pathogen genomics, I want to make the case, is a discrete discipline from human genomics, and for good reason. So in pathogen genomics, the primary focus is just a single species in humans, obviously, but whereas in pathogen genomics, we're responsible for a whole range of different species. The scope is broad: viruses, bacteria, and fungi. In addition, the application is also different. So we're, as I mentioned earlier, we're responsible for initial pathogen diagnosis and as well as population level surveillance and outbreak response. The scale and timelines for us can be large volumes, and it can be real time or near real time. So in the space of, as I mentioned, days, and certainly not weeks in terms of turnaround times. And the genome characteristics vary depending on the pathogen that we're looking at. So there's multiple sample types. Some are RNA, some are DNA, some have single strand, some have double strand, and so there's different things for us to look at there. And of course, evolution is acting on all pathogens, so it's highly dynamic, fast evolving, and as you will have heard in Shannon Russell's talk 2 weeks ago, can involve horizontal gene transfer and as well as recombination and other things that can confound the sequencing data too. So there's a lot to keep on top of there, and in addition, the analytic methods are different, and the pathogen, the bioinformatics piece, which I'm responsible for at PHL, is a discrete discipline in and of itself from other disciplines in bioinformatics. And of course, we have integrate. In addition, there's a need to integrate the results that we get into clinical records and epidemiological records, as well as other data like lab metadata, case and cluster tracking. And we have a whole range of end users, as I've already mentioned, and these need to have this data provided to them in different ways, specific to the end user and their particular needs. And then, as Michael mentioned, I came from academic research, and when I came to the PHL, it did feel like sometimes you're kind of getting hit by a fire hose sometimes, and this fire hose never gets turned off in terms of the amount of data and information and things that are generated. I just present this as a means of, not as a means of I think just as a means of illustrating that there's a difference between academic applications of genomics and public health applications of genomics. So in public health, we obviously have this constant stream of samples, and this can't be turned off. We can't pause it just to wait for figuring out what's going on in something or to analyze something. We've got to be able to continually analyze the products that we have in operations. Turnaround times are important. There's different, as I mentioned, different types of analysis, wide range of pathogens in the academic environment I came from. I was focused on a very specific bug and parts often only of that very specific bug, whereas we need to maintain a very broad and shallower understanding of the pathogens, but still deep enough that we can understand them properly. We have validation and accreditation requirements as well, that particularly for any clinical test. So those are different. And the piece that I think that people don't always, where I come from, but always necessarily grasp is that data integration of the genomics data is not easy at all. It seems simple on the face of it, but it is probably the thing that takes more time and more work and presents more challenges than almost anything else. And then, of course, we've got to be able to translate the results to a range of stakeholders, including physicians, epidemiologists, and then they could be policymakers, people who don't have genomics backgrounds, the public as well, and other places. So there's a whole range of different sort of things going on in an operational public health lab as compared to maybe necessarily an academic lab. Okay, and so genomics at the BCCDC PH pre-pandemic was a quite different landscape, but I think a really essential development ground to get us to the point where we are today. I started in the PHL in 2021, so this predates me, and a lot of the genomics program development predates me, but this has been the essential foundation that really allowed the lab to build off that during COVID. Pre-COVID, there was a small amount of sequencing capability, and it was operationalized. Some of it was operationalized as part of PulseNet, where enteric outbreaks are investigated. There was some CPO genomics and some CPO provincial surveillance, but there was limited dedicated staff available to that, and throughput was low. So up to 24 samples a week previously, and it could take 3 to 4 weeks to generate a whole genome sequence result, and perhaps months to generate an outbreak report. And then so during COVID, and as part of this development, the lab developed a functional module structure. And the purpose of this, and this is, I think, really key to how we've been able to roll out pathogen genomics in the public health lab, is that it's pathogen agnostic. So it allows us to respond to and develop new sequencing products rapidly without being siloed into, say I work only on viruses, or I work only on bacteria. So because we work on all of it across the structure, we're able to take a reasonably pathogen agnostic approach. Each module is led by a team of medical, clinical microbiologists and senior scientists, and there's expertise overlap between them, and close collaboration. And it starts down the development end with an assay development unit, and then goes into wet lab genomics, the bioinformatics, and informatics, which is integration of the genomics results. I just call out Chris Riel, who developed the Plover database, which is our genomics database during the pandemic, and was able to serve information to the public, not to the public side, but to health authorities and to people like contact tracers, and then the translational support provided by Shannon Russell and others to all our end users. And then this is just a snapshot of where we're at, and this is out of date by about a year. But we have published many sequences on GISAID of whole genomes, and we've investigated thousands of enteric organisms, more than actually 600 CPO plasmids sequenced, produced a large range of cluster reports, populated dashboard views for others, and also I have mentioned wastewater surveillance, but that's another piece. So this is just an overview, and so now I'm going to hand over to John, who's going to give an insight into the SARS-CoV-2 genomics.
Okay, thank you, James. Yeah, so we're just going to go through a few examples now of kind of where we are with certain pathogens, and kind of some of the approaches we take, and kind of where we're going with things as we move from, I guess, the foundational kind of work that when I first arrived around SARS-CoV-2 and the genomic approaches we took with that, and then how we're translating that through into other organisms now and other pathogens. So with no intention of giving anyone PTSD around SARS-CoV-2, I'm not going to do too much of an introduction on this. Just to say that, we first started standing up our genomics on SARS-CoV-2 around March 2021, and that's when I came down to trying to help set up the first kind of genomic sequencing with SARS-CoV-2 at BCCDC, and that was done on our nanopore platform, so relatively low sample numbers at that point, because the lab was primarily focusing on standing up its LDT assays and things like that for diagnostics. And that eventually translated through to the end where we went fully operational. And as James says, we've ended up doing, upwards of 200,000 sequences that are now publicly available. Now, the purpose of that are numerous, and it's kind of evolved with time. Initially, it was focused around transmission networks and outbreak investigations to try and see, where things come from, where could they be linked to, were they imports, were they local transmissions and things like that. And that kind of moved into things like, variants of concern and monitoring for that, because obviously the sequencing primarily is, relatively agnostic to any changes, and you can easily see that. So variants of concern could be spotted. And then a lot of work through groups here at the BCCDC focused on looking at vaccine effectiveness and using that to see whether there's any lineage differences in terms of what those kind of effects might be. And now going forward as well, we're looking primarily around, we do onward sequencing. It's changed in terms of the frequency that we did. We do, I guess, in a relatively small amount now compared to what we were at once doing. But that's still to monitor lineages that then helps us kind of inform on vaccine inclusion for ongoing seasons. Things that are coming online now as well, and we've kind of initiated things like wastewater monitoring around March 2023 with wastewater groups, and obviously work with Natalie Peschecki and things like that here at the Public Health Lab. Because what we're having to do now is, obviously our clinical testing has dropped significantly, and in order to understand, say, the population dynamics of what's going on outside, things like wastewater monitoring gives us a window into a full population study, which we don't necessarily see now with, say, acute clinical cases or the relatively low number of samples that we get. What I want to do is go on to the next slide, James, if you can, just to point out kind of the approaches we take. And this is illustrative actually of a number of kind of pathogens that we undertake to sequence. When we understand, so when we have the sequence of a known pathogen, we can actually design primers to actually do tiled amplicon generation across the whole of the viral genome, and we generate these multiplex pools of products, which are then sequenced. And we here at the lab, we use two approaches. One is our clinical WGS, where we take those pools, they're mixed together and then sequenced on our Illumina platforms. And that's done with pieces of around 1,200 base pairs. So this is relatively kind of, 29 pieces across the whole genome, and we sequence that to give us the information. We also have an approach which uses higher resolution 400 base pair fragments, i.e. 96 fragments across the genome. And that gives us greater sensitivity. You can see there at the bottom, at CT less than 30, the 1200 scheme holds up. Whereas when you start getting into high CTs, that 1200 scheme falls off a little bit, and we need the smaller fragments to gain us sensitivity in samples that may be degraded or higher CT values. With that sensitivity, there are some caveats that come with that, and that's around the primers and whether you accumulate mutations and whether we see dropouts in the data. James, you want to go next, please? And this is just to point out, as a resource with time, as the virus has evolved, and this applies to other pathogens as well, there's been a need to repair these amplicon schemes. You can see on the left-hand side there the vertical red lines, and that's an indication in those particular samples, which are arrayed vertically. There's been a red dropout amplicon, so we're losing sequence information in there. And what you have to do as time progresses and you accumulate these mutations, you have to repair these things to give you back complete data, and that's sort of illustrated in the middle there. And then at the end is our full kind of production amplicon scheme with a 1200 size. This is just to point out that actually this is an ongoing effort, and we maintain these schemes and not only use them ourselves and maintain them for ourselves, we also make them freely available internationally and nationally. And we make that information available so people can actually go to a source and have the information to be able to do this themselves. We do the work and to put the resources into kind of creating these and to maintain them. So actually it makes it easier for other people to be able to use those. And this is an ongoing international effort with, then we like to say, the Arctic Network in the UK, which is focused on a number of different pathogens. Go next, James, please. Just want to point out that obviously with the changing landscape, obviously at one point we were doing a very large amount of COVID sequencing. Now that's not so much. We're probably around 70 to 80 every 2 weeks, and that lets us keep an eye on the circulating strains. But what that doesn't necessarily do is give us that same high resolution lineage across the population of what may or may not be going on. And this is where our kind of wastewater sequencing has come in. It gives us that population view. But it's just to point out here that you can't just have one or the other. Without some clinical sequencing, you can't interpret the wastewater data. We need to have the ability to, identify individual lineages, so we can then assign those into our wastewater. So just to say, it's a combination that gives us the power. We have our overall population surveillance, for example, with the wastewater, and then by having clinical data as well, we can even look at the deltas between the two. So if we see a skew in the representation of the lineage between clinical kind of severe cases and then our overall population lineage profiles, then that may be indicative of something that's becoming of more clinical relevance or a severity measure. So we have a dynamic of changing landscapes, and also we're trying to move with that. And there's one more thing I just want to illustrate, James, if you can move on, as a thing we're seeing now, and it's quite interesting, and it's just, again, it's something we can see within the sequencing data, and that's the issue of chronic infections or ongoing infections in individuals. Obviously, we're used to say having catching COVID, and then we have it for 2 weeks, and in the vast majority of cases we recover from that. Now, in some individuals where we have immune compromised situations, you can see here we can pick up individuals that have had infections for actually quite long times, over a year, and I think where we are currently, we've seen one around 2 years now. So we have these situations where are we trying to kind of help out in terms of identifying these individuals? Can we trigger any kind of increased kind of therapy, like interventions medically? And these are kind of the things we can only see from sequencing, and we can pick these up based not just only on having multiple samples from an individual, but we can also find like a context. The top diagram there, you can see that's our kind of, if you want, the lineages that we've seen over time from 2021 to 2022. And what you find is, given lineages are very kind of finite generally in terms of when they're around, and then we evolve into the next lineage and the next lineage in terms of the name as the virus evolves. And these chronic infections actually can show up very easily for us because we have a lineage that's completely out of context temporally. And if you want to go just quickly to the next one, James, I can illustrate that. Each one of these, you can see here the different lineage names, and see the top kind of panel shows you that's the kind of the numbers of that particular lineage we had with time. You can see really pretty much, they're very kind of a normal curve over a given time window, and you can see it. But what you see is those punctate ones later on in the timeline where you can see an individual pickup. That lineage comes back, if you like. So we can spot these kind of lineages, and this is only information that we can get from the genomic information and our sequencing and our database, things like that. I'm going to hand back to James now for another example, things we're up to.
Thanks, John. So I thought I would talk a bit about influenza. So this is obviously front of mind where we are now in terms of both flu season that we're sort of in, and also the avian flu panzootic that we're experiencing. And flu has obviously been historically a significant threat to humans. There's been a range of epidemics and pandemics caused by influenza, and so it's another important pathogen for us to be able to keep tabs on and to be able to respond to quickly. So BCCDC PHL began sequencing influenza in 2022, but we also, in addition to sequencing seasonal influenza, we sequence samples from the Ministry of Agriculture as part of a collaboration with them to sequence wildlife samples. And this is important because influenza is an interesting virus, but one that has obviously a lot of interaction with animals, and that is something that we have to worry that interaction between seasonal flu and avian influenza or other types of influenzas, we had swine flu previously, could result in another human epidemic or worse pandemic in the future. So this is useful, a really useful collaboration that the lab has with the Ministry of Agriculture. And so I want to highlight that. And I also just wanted to highlight that influenza is a different type of virus in some ways from COVID. So I should call it SARS-CoV-2, which is a scientific name. But John, what John was just talking about that, and that is a single-stranded virus. Influenza is a segmented virus. And what that means is that each separate piece of it has 8 separate pieces of genome that can sort of be thought of as like individual chromosomes, if you like. But the important point about them is that they're sort of like modular building blocks, and they can be swapped in and out and with other segments from other influenza viruses. And when that happens, that can result in a novel reassortment. And it's that in particular that we worry about in terms of emerging threats in influenza. Typically, in the past, a lot of the focus has been on, and rightly so, the HA and NA proteins, which, as you see, they labeled there on the surface, and these are the antigens that interact with the human cells and the vaccine targets. And so those are important. Flu is subtyped and classified based upon those sequences. So the subtypes H1 and N1 and H3 and N2 are the currently circulating seasonal subtypes of flu. But in addition, we have now this H5N1 subtype that is circulating as part of the current panzootic in wildlife. And I just wanted to just show the difference between traditional sequence analysis and whole genome sequence analysis. So in influenza, a traditional sequence analysis would have PCR'd up part of that HA segment, and then it would have used Sanger sequencing to produce a consensus sequence. And so what that means is that you're getting part of the HA segment, and you're getting a sequence read depth of a maximum of 2, where those 2 forward and reverse reads are lined up. Whole genome sequencing, on the other hand, allows us to sequence the entire genome, and it's relatively straightforward in terms of like how we extract and PCR up that genome, and can use a small number of primers to do that. But we get all of the segments, full-length segments now, and we get much higher depth, and I'll come to why that's helpful in a minute. But just to illustrate why like the traditional approaches are limited in their resolving power, this is a phylogenetic tree and this is real data of HA samples. And you can see in this tree that on the bottom is the divergence, so how much they differ from each other in terms of nucleotide differences. And so it's a very small divergence window there between 6 and 14 nucleotides. And you can see they're lined up on those lines a lot of dots, which are on the same line, which means that they have the same sequence, and we can't split them apart just on the basis of the HA segment alone. But if we instead build a phylogenetic tree from all 8 of those segments, and so basically by sticking them together in the same order in each sample and then building the tree from there, you can see much more diversity. And in fact, you can see clusters and groups down at individual levels there. And this really helps us to get a better understanding as to what's going on in particular group of samples of influenza. And we can stick these trees next to each other, and so this is called a tanglegram, and you can see here that not only do you get much more diversity on the concatenated tree on the left, but all those intertangled wires connecting them show that actually the picture on the right, the HA segment, is moving about depending on what the final result is based on the whole genome. So whole genome sequencing really does allow really high resolution for influenza. So the current panzootic from avian influenza began in 2020. The first BC detection of an H5N1 virus was in 2021, and it's really involved an unprecedented amount of spread across the world and transmission into mammals as well as other birds and other animals. And to date, there have also been a very small number, but some of them significant, of human infections resulting from these. As part of this, I didn't know anything about birds, but I had to learn at least a little bit about birds. So migratory birds fly on flyways, and they migrate up from the south in the spring up to Alaska via these different flyways in North America. And you can see here that we are on the Pacific Flyway, and then there's also the Central, the Mississippi, and the Atlantic flyways. And when they migrate up north in the spring, then they intermingle often with birds from other continents as well, and then they come back down here in the fall, back down the Pacific Flyway, back down to the southern hemisphere, and they land often more in the fall than in the spring. They land in the lower mainland, where it's seems to be a particularly attractive landing site for them, and this is. As part of the H5N5 panzootic, has caused a large number of both dead wildlife and infected poultry farms as well. And so one of our concerns is that the lower mainland in BC has the potential to be a sort of ground zero for new emerging influenzas that are resulting from this. So it's really important that we keep this the surveillance ongoing. In the avian influenza outbreak, there's been a classification scheme developed so that those individual combinations of 8 segments are classified, and that classification produces a genotype. And so you can just see here on this phylogeny, which is a much larger phylogeny of a lot of things from the ongoing panzootic, that there's a range of different genotypes that are causing infections, and we have seen waves of these come. The most recent wave is caused by genotype D1.1. You'll have also heard of large outbreaks in cattle in the US. Most of those to date have been caused by a distinct genotype B313, although recently there were some more D1.1 cases there as well. The US cattle led to a number of human infections, most of which seem to be mild, but there have also been infections in humans caused by D1.1. And of course, you will also be aware that one of those was in BC just about this time last year. I just wanted to give you some insight into that and the sequencing and how that was done in the lab here. I remember, because I remember this very clearly from the time. So on the sequencing on the sample really started around Tuesday morning, where the amplicons were PCR'd up and gotten ready for sequencing. And I went up to see John in the lab at about 3 o'clock, and he was just literally loading those onto one of the long read sequencers that we have. They were also loaded onto the short read sequencing, which we use more operationally at the same time. And then I got home, and John sent me a text saying that the long read stuff was ready for analysis. So I analysed that at that time, and within 2 hours of that, we had an initial result of H5N1 being confirmed and the specific clade genotype being confirmed. And so within a day, we're able to get that information out quickly. This was reported back to the program heads at BCCDC and then up to the provincial health officer very quickly, and we sent those sequences to the National Microbiology Lab, who also have an important role in influenza, and they were also sequencing this virus. They got back to us the next morning, confirming that their results were identical to ours. And then by Wednesday, we had the short read sequence results from our operational sequencing, and then by 1:20 on Wednesday afternoon, we had the initial phylogenetic trees, which really enabled us to contextualize where this sample had come from. And you'll probably be aware that we still don't know exactly how that patient acquired their infection, but the infection they had was very severe, and it was very important that we had an understanding as to where that came from. And so you can see on this phylogeny from the publication that we were able to group those samples, that individual sample, alongside near but not exactly the same as other wildlife samples, and they were somewhat distant from some of the infected premise samples, and that was important because it meant that it was not necessarily coming from those infected premises where there were farmed poultry, but probably from an infected wildlife sample. So we were able to do that really very quickly. And then just to flag that whole genome sequencing also really allowed much more than just the sequence and just the genotype. And you have to know that when someone is infected with influenza, the virus spreads and then they get sampled, and then what you're sampling is a population of virus. And that virus can in fact have different sequences. If some of them have mutated, then it can have populations of sequence types. And in this example, I'm giving an example where there's a mutation in the PB2 segment, and so some of the viruses have that mutation and others don't. And so if you just said, when you look at the genome sequence, if we see any mixed bases and we just ignore those because we're not sure why they're mixed, you would miss this. But that's not what the sequencing that we do does. The sequencing we do does reveal that actually in PB2 there was a mixed base, and this is a script that was developed by one of the genomics specialists to pass that information out. And you can see at this specific location in PB2 that there was a almost 50% mixture of these two nucleotides, and that was leading to this E627K mutation that can enhance mammalian adaptation. We did also find other mixtures as well, and so it's just a flag really that the WGS can allow much more than just the initial typing, but also information about things like mutations that might be useful. And then so I just would finishing with avian influenza. I think that this is I asked AI to come up with an image illustrating a bird with a flu virus tapping at the window. I think this is sort of where we're at with avian influenza at the moment. It's not yet blown up into a huge human problem yet, but it has affected some people very seriously, a very small number of people, but it has done that, and I think has the potential to be worse. And so I think we just need to be careful and vigilant for this. But I also want to highlight that our 3 years of experience as a team with the Ministry of Agriculture was really critical for enabling this rapid analysis and contextualization at the time. And I think WGS will be essential if we have more cases. And so just to acknowledge the team, particularly Shannon Russell and others on the team who worked really hard to provide the flu analysis and surveillance to the Ministry of Agriculture and others. And so now I'm going to hand back over to John. I think he's going to talk about measles.
Yep. Go ahead, James. Next one, please. Okay. Thank you. Yeah. Just trying to run through another example here and then show you some of our products that, in addition to what we've already described, obviously fairly topical at the moment is the measles and our measles outbreak and the and Canada's loss of elimination status. What measles generally is done when samples are sent through to the NML and they're typed using an N450 region, which is the very beginning of the genome. It's a 400 base pair fragment, and you can see in those trees down the bottom right there the measles N450 virus, and this is some of our BC samples, and you can see actually on that tree there's a very large vertical line there of individuals that would show no difference at all in that sequence. Whereas when you go to whole genome sequencing, which we've stood up now for measles itself, you can see that tree on the far right there becomes much more elaborate, and we can actually see connections and kind of start to interpret some transmission networks and some differences there. So it's just to illustrate, the utility of kind of the whole genome sequencing situation for this particular product, and we're kind of determining now, where we're going to go with this and whether this is something we need to be doing or not currently because of that loss of status. And these sort of things are also potentially something we could be looking for in wastewater as well. And if you can go next, James. And that's just kind of a part of a portfolio of things actually that we now run here at the BCCDC, coming out of that early days and those resources that were put into the SARS-CoV-2 pandemic. Obviously, what we're trying to do now is utilize those resources and make use across our full portfolio of pathogens, use the sequences. Obviously now we're not doing SARS-CoV-2 sequencing, but what should and do we want to be using that infrastructure for? And you can see here there's things we have in operations. Obviously, we still have SARS-CoV-2 both clinically and in wastewater. And as James has said, we have flu sequencing. We do mpox sequencing as well. A lot of bacterial stuff, obviously where we started originally, under Linda's kind of guidance with the PicNet and those kind of surveillance kind of operations. We do CPO, TB is a big program we have here. And we have various platforms for that, both on the Illumina and the nanopore systems, which I'll come on to in a sec. In operational development or on hold, we have things like flu, like I said, measles, RSVA, RSVB, some syphilis, AMR work, as well as kind of standalone nanopore sequencing to get full bacterial complete genomes and plasmid sequences. In development, more on the research sort of side, we also have programs around things like syphilis, WGS sequencing, but also emerging pathogens and works beginning to happen around metagenomics and, how do we find the new thing that arrives, not just what we already know. A lot of these schemes that we design are prerequisite, so you have to have a prerequisite of actually having a sequence to actually design to. Metagenomic approaches obviously are agnostic to that, like similar shotgun sequencing approaches. They don't have the sensitivity, they're a discovery kind of focus, and then we migrate into some of these amplicon scheme type approaches once we have information that we've gleaned from metagenomic approaches. Go next, James, please. Just want to point out, obviously here at the BCCDC we have a number of different platforms, primarily focused with Illumina short read sequencing, and then nanopore long read sequencing. Obviously they use different chemistries and the different approaches. We have large operational capacity on the Illumina side and relatively large capacity as well when we need to on the nanopore side. We also kind of pick and choose depending on what we need and which approach we'll take, and I've kind of alluded to that a little bit. A lot of our high volume virology type stuff is still done on our short read platforms. The long read platforms come in around things like CPO and understanding plasmid rearrangements, and when we need high structural kind of understanding of what's going on, which we can't discern with some of the short read approaches. We can go next, James. Thanks. This is just kind of illustrating the point of I guess this is more of a future view or something. We're trying to understand how genomics is going to be integrated into our kind of our system, or we at the BCCDC obviously are in the fortunate position and we have a lot of high capacity and resources to do these things. Obviously we realize that hospitals and remote areas are obviously wanting to be able to do this if it makes sense from turnaround times and that sort of thing. Where things become a bit more of an issue is around the resources for analysis, integration of the data. So it's a kind of a two-way kind of understanding and a two-way relationship we want to try and build and try and help people get to where they want to be and what the system kind of needs us to kind of where we need to be. One thing I would point out is actually sometimes realistically the bottlenecks now are not necessarily the actual sequencing, as James has mentioned before. A lot of the kind of upfront sample logistics, logging, sample processing are substantial kind of things to have done and get done, and we've benefited amazingly from actually that integration here at the BCCDC in terms of that sample receiving, sample processing and the system, as well as that data integration from having the BCCDC kind of epi side of things here located in the same building. And I would just like to point out one thing in particular is scaling and centralizing onto larger and larger and larger systems doesn't necessarily always work when in situations where we have demanding, say, turnaround times. We can't afford necessarily to batch onto massive systems all the time because we have kind of constraints on the turnaround times that we need to provide. So there is a kind of, I feel like, a Goldilocks zone of efficiency, and bigger is not always better. And a good example of that was with our COVID work. These systems generally have a certain number of samples they have a capacity for, and we had to do a lot of work to get expand that capacity and those custom barcodes that we use for tagging samples to make it economic to actually get these systems to run, because our viruses are very small. So it's not like human genomes where we have relatively small numbers, large data for an individual sample. We have very large numbers and require very small amounts of data to do the analysis that we need. So it's very much predicated on what do we need for a given situation. You're going to go next, James. Thanks. This is just to point out kind of, I guess, where we came from and where we are now in terms of the pathogens that we have, online now. And obviously work goes on as well to try and incorporate new pathogens. And there's an expanding range of priorities that we have, and be that to inform patient care around TB and HCV, our clinical testing and validate pipelines and analysis workflows. Also essential things for our surveillance that we still provide around SARS-CoV-2 lineage monitoring, so people are aware of what's around, and also the turnaround times, like we've spoken about, 3 to 7 days, where we have WGS result going on from that. I think go next, James, please. And this is just to point out as well before we get towards the end is we try and be as open as we can with our data. I mean, we've shared, upwards of 200,000 genomes of SARS-CoV-2. We share other kind of pathogens and information like mpox and those sort of things. And this is an ongoing discussion with us on how we make that publicly available within our constraints. And where we can as well, we're often providing the bioinformatic pipelines that we use that people can actually use themselves, and also around our wet lab protocols on things like protocols.io and things like that. Okay, thank you. Next. And this is an ongoing development, and TB is a great example of that. Where we've got now clinical validated pipelines in place that have come out of this work. And this is actually we were actually one of the first accredited labs in Canada to have this set up and in clinical use. Next, please, James. So the future, I guess this might be it gets a bit speculative and a bit in your face, but as more and more kind of pathogens get worked up, our monitoring systems, the more and more we can get these things integrated. This is becoming a great tool in our armory to try and provide some real-time monitoring, and we're hoping to try and speed up this kind of turnaround and this integration of all our strings of data to allow us to give these early warning kind of systems and facilitate kind of information feeds to the relevant people, be that our medical health officers and people like Linda and our medical directors. And so we can integrate this data into kind of our system. Thank you. Next. And again, the final acknowledgement, obviously this is a huge amount of work performed by many individuals, many listed here, but I just want to give a huge shout out to actually the individuals who aren't listed here on the public health lab, our technicians and all our different labs. I mean, they've put a consistent, high quality, highly dedicated kind of amount of work as individuals into this, and I just want to give a good shout out to them. We talk about it, but this doesn't happen without a very large and considerable effort from a very large number of individuals. So I'll end it there, I think, and then if we've got any questions. Thank you.
Thanks very much, John and James. James, you got more to say, or?
No.
Perfect. So again, thanks very much. Very interesting. I know I was involved in a case, and it was to do with mucor and fungus doing a surgery frozen section. And I called them up and said it looked like mucor, and they said, "Oh yeah, we know it's rhizopus," because I think they'd already done the genomics, which is important. Anyways, questions for John and James, Dr. Gao.
Well, yeah, it's too sophisticated for me to ask any questions. Just maybe a few comments. One is this is amazing work, and we always talked about at BCCDC about precision, personalized medicine. But this is precision, more of a precision public health. Is actually much more impactful and help to guard the health of the entire population. Thank you guys for your great work. So I want to utilize this opportunity to emphasize one of the announcements we made. So just a day ago, Genevieve has forwarded my email to everyone. So the government of Canada has invested $1.7 billion for Canada Global Impact Research Talent Initiative, which has like research chairs funded by $1 million a year for 80 years. And then $500,000 for another 4 years, and emerging leaders is $100K a year for 12 years. So this is a huge funding initiative with CFI support and everything. And so for anyone, you guys know that if this is for recruiting international talents, particularly from the US. If you know anyone who is qualified, please ask them to send the CV or to Genevieve and register the link that Genevieve will send it out. Particularly relevant to today's talk is actually one of the funding priorities from the federal government is the food and water safety. So among is digital health, digital technology, health, biotechnology, but there's like a food and water security is one of the focus of priority funding from this particular initiative. So it might be relevant to BCCDC for getting an internationally renowned person actually recruited and here to lead some of the and the great things is already happening here. So I'll stop here. Thank you.
Thanks very much, Gao. Linda?
Hi. I'm just so impressed with these guys. James and John, as well as Chris and Shannon, our genomic scientists, have been really critical in responding to public health pressures and emergencies. And I appreciate Gao bringing that up because a few weeks ago, BCCDC had hosted a global health forum down at as part of our research week, and it was an international forum with WHO representative and folks from the US and others. And one of the things that we had talked about is global health security and how genomics really is, pathogen genomics is really critical for that. So I'd love to follow up with you if there's opportunities for resources for us to do that work.
Yeah, particularly to recruit somebody like internationally renowned to our place to work together with you guys.
Yeah, sounds good.
Thanks, Linda. So again, James and John, thank you very much. Are there questions or comments for James and John? Well, thank you very much, both. A very interesting talk from my perspective, and enjoy the rest of the day and enjoy your vacation over the next little while.
Okay, thank you.
Thanks, guys.
Thank you.
Bye-bye.